Hi, I'm Heather James, conference producer at Technology Networks, and I'm here to introduce our next talk, Deciphering Metastasis through Circulating Tumor Cells Research, from single cancer cells to their derived advanced models. I'm pleased to welcome Dr. Sara Abalde-Cela. Dr. Abalde-Cela is the research group leader of the Medical Devices Group at the International Iberian Nanotechnology Laboratory in Braga, Portugal. The Medical Devices Group focuses on the development of optofluidic platforms for the diagnosis and understanding of diseases. Sara holds a PhD in nanotechnology by the Universidad de Vigo and a postdoc in the field of microfluidics at the University of Cambridge. A very warm welcome to you, Sara. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask your questions using the Q&A system to the right-hand side of the video player. And without further ado, I will now hand over to Sara to begin her presentation.
Thank you very much for the introduction. It is a pleasure to be here to be able to present our work, as Heather said, on the deciphering metastasis through circulating tumor cells research. So without any further ado, because you already know my affiliations, I would like to explain what we do in our research group. So we are focused on enabling precision medicine through accurate diagnosis using key enabling tools. Some of these tools are based in microfluidics, and with these microfluidics, we isolate biomarkers, liquid biopsy biomarkers that I will dig into it a bit later in my presentation. And once we have these biomarkers, we can analyze them with traditional technologies such as fluorescence, NGS, etc. But we can also go a bit beyond these gold standards and traditional techniques by using nanotechnology and nanobiosensors to offer a quantification of these biomarkers in a multiplex way. Putting together these technologies, we can also explore advanced models in order to achieve devices to understand disease processes. And finally, we also integrate these type of approaches to produce disruptive optical instrumentation and medical devices. So today, most part of our research focuses on metastasis and on the hallmarks of metastasis. And more specifically, we've been trying to provide tools for the activating invasion and metastatic processes related into the microenvironment and also into circulating tumor cells' role into these metastatic processes. Because as we all very well know, metastasis is accountable for 90% of cancer-related deaths. So the way metastasis happens is we have a primary or an active tumor, and some cells are able to escape these active tumors through circulation and then colonize secondary organs. This colonization is related to many different factors, such as motility, invasion capability, and the microtumor environment status. So, in this talk, we will mostly focus on single-cell analysis and tools to provide this single-cell analysis, because when we do bulk cell analysis, as tumors are heterogeneous, we might be missing some individual properties of those individual cells. Hence, it is very important to be able to provide differentiated expression profiles for these single cells to be able to analyze cell subpopulations and, for example, identify which cells have a higher capacity of adapting to the tumor microenvironment or a higher colonization capability. Putting it into very simple terms, we will be trying with different tools to assess which one is the one malignant cell and how much is this malignancy potential, and then all the factors that might be associated to this malignancy, abstract terms such as, for example, relation to overall survival of patients or other critical clinical uses. Though there are some limitations in the current standard single-cell analysis, which might not be sensitive enough, might not be scalable or accurate, and there is also very difficult to be able to reconstruct a clonal evolution. And so to be able to provide these tools that overcome these limitations to then relate into immuno-oncology developments, it is very important for the field of metastasis in particular. So as I briefly mentioned, the monitoring of cancer as it is nowadays in the clinic is invasive. It cannot be continuous or reliable because it is done through imaging technologies mainly, and these imaging technologies might be expensive. As I said, they are invasive and also cannot be made as frequently as possible. So they often miss the early spreading of the circulating tumor cells, CTCs for short, and micrometastasis. So it is very much a need to develop these non-invasive, continuous, reliable, and tools that provide real-time information on the cancer status. For that, I will be speaking about liquid biopsy, and liquid biopsy is a field that explores circulating biomarkers in body fluids. It can be very different types of body fluids. Mostly there have been studies in blood, but also it can be in urine, in spinal fluid, or in other types of body fluids. The most known circulating biomarkers would be circulating tumor cells that I have already mentioned, and also circulating tumor DNA, as well as extracellular vesicles. Today, I'm going to focus my talk on the role of circulating tumor cells into these metastatic processes and how those can be used for monitoring, prognosis, and diagnosis of cancer. Now, one of the challenges for circulating tumor cells is that they are very rare in circulation and at very low concentrations. So if we draw blood from a cancer patient, we're going to be getting billions and millions of red blood cells and white blood cells. And in the middle of all these millions and billions, we're going to have a few circulating tumor cells. Now, being able to distinguish or eliminate the red blood cells from this complex sample is fairly straightforward and easy. However, because white blood cells overlap in size and in some other properties with circulating tumor cells, it is a challenge. It has been a challenge in the field of liquid biopsy. So it is like finding a needle in a haystack to be able to access the CTCs to then further study them and their influence on the metastatic process. So there's two challenges here. I have mentioned the first one, which is finding this needle in the haystack, which is related to sample processing of these complex body fluids. And then there's also the challenge of the sensing technique to be able to provide information about the characteristics of these CTCs. So going further into the sample processing bit, today I will present some solutions that take benefit of microfluidics technology. And microfluidics is basically a technology that uses microchannels to process fluids or to process body fluid samples in this specific case, and is able to contain small volumes. We are able to control spatial and temporal, the behavior of these fluids. We can provide a fast and automated analysis, as well as miniaturization and integration. And it also has a low cost because the fabrication of these devices can be scaled up easily through standard plastic processing product development fabrications. So just digging a bit into the problematic of the metastasis, besides being the higher cause of cancer death worldwide, it also has a very high impact in a socioeconomic profile of the different countries. So, for example, in the US, $150 billion goes spent in cancer annually in cancer treatments. However, 75 of these treatments are non-effective because, as I have mentioned, cancer is heterogeneous and it is also dynamic. It changes with time. So it is very important to have tools that are able to assess the cancer status at a specific time points of the course of a cancer in a specific patient. Now, as I briefly mentioned, the problem with the usual techniques being, as I said, imaging technologies here on the right of the slide, or tissue biopsies, is that tissue biopsies, despite offering molecular information, do not reflect the heterogeneity of the full material in the tumor, and also they're invasive, and they do not allow this spatial and temporal monitoring. Then with the imaging technologies, we could do continuous monitoring. However, we know it is expensive and it also exposes patients to continuous radiations. It is not sensitive because it often misses micrometastasis. Hence, we cannot do early detection, and it does not provide molecular information, unlike these tissue biopsies. And that is why on a metastatic setting, often treatment selection is based on outdated information that is coming either from the tissue biopsies at the first diagnosis point or from the imaging technologies. Now, a liquid biopsy can, through the analysis of circulating tumor cells, have the potential to alleviate these shortcomings because it is non-invasive, it allows for this early detection, provides molecular information at many different levels, at the proteomic level, but also at the genomic and transcriptomic level, and it allows continuous monitoring because it basically relies on these body fluids. However, traditionally, the liquid biopsy technologies devoted to the isolation and analysis of circulating tumor cells haven't been sensitive enough because they miss a lot of the needles in the haystack. There's also no standards in the clinic yet, and there weren't many proofs of clinical utility up to very recently. Hence, our research group has spinned off a startup that is called Ruby Nanomet. And so the first part of my talk is going to be devoted to show you the advances that we can have with a specific microfluidic device that we call the Ruby chip to be able to isolate and analyze the circulating tumor cells. The way this solution works is with a draw of a blood sample of 7.5 milliliters. We can just run this whole blood without any further pre-processing through what we call the Ruby chip device, that is a disposable unit that provides very efficient isolation of the circulating tumor cells. This isolation and this process of sample processing is automated with a unit that provides on-site processing, staining, and image acquisition, and then a further analysis of the cells and their characteristics based on fluorescence markers through immunocytochemistry, and then provides a full report to the oncologist with a classification and subtyping of these different cancer cells that can then be related to druggable targets. So with this fully automated solution, we ensure no inter-user or inter-laboratory variability. And this is something that, as I mentioned, has been lacking in the ecosystem of circulating tumor cell isolation and analysis. So this would be the value proposition, and we actually have demonstrated. You're going to see some examples that we can detect metastasis and progression up to 1 year earlier of current technologies. And this technology has been also applied to 10 different types of cancers. Our current sensitivity is 97% and specificity 100%. And we can do, for example, simultaneously with very fast sample to result of 3 hours. So I'm going to start from a very simple comparison and optimizations of the microfluidic system. So first of all, one of the largest challenges, as I mentioned, is that because CDCs overlap in size with the white blood cells, it is quite difficult to have a depletion of these white blood cells. Our microfluidic device is based on size and deformability. And so because white blood cells have a smaller nucleus, are more deformable than cancer cells, hence we're taking advantage of these size and deformability differences of the CDCs versus the white blood cells. So we have reached up to 70% isolation capability of the circulating cancer cells using spiked samples. So that is blood from healthy donors that has been spiked with cells from culture. And then we have a very good white blood cell depletion, still with 10% CDC purity, which is still much higher than that is reported from other technologies in the field and in the literature. Now, more interesting, if we move towards real samples, in this graph on the left, what you can see is a small cohort of 9 colorectal cancer patients that has been tested with the Ruby Nanomet technology versus the cell search technology. So cell search was the first and only approved FDA technology for the isolation and characterization of CTCs in the context of breast, colorectal, and prostate cancer, and has been the gold standard in the clinic for several years. However, cell search, due to the basis of its technology, is not so sensitive. And as you can see, if we get two samples from the same patients and we run them blindly through either the cell search technology or the Ruby Nanomed's technology, you can see that the sensitivity, so the number of cells of CTCs that we are able to capture is much higher for most of the patients. Besides, we don't need sample preparation. We can isolate all the phenotypes. That is very important because cell search is based on the isolation of cells that express specific markers. However, because we use this size and deformability, we're agnostic to the protein that is being expressed in the cells, and then the proteins of interest that might be druggable proteins can be put into the assay by choice of the oncologist. So it allows phenotypic profiling, and also more important in the context of immuno-oncology for therapeutic targeting, it is that we have viable CTCs, so they can be used for other types of downstream analysis, included functional studies. And also it allows patient stratification, as you might see in the following slides. Now, going even further, increasing the cohort and increasing the number of types of cancers, I bring here another example. It's again a benchmarking study against cell search. In this case, we have 30 samples from metastatic breast cancer patients. It is 15 patients and 2 collections at different points, the first point of diagnosis and at a first follow-up after treatment. And in here, what we can see is again that with cell search in yellow, most of the patients have 0 or 1 CTCs. Only a couple of patients have more than 5 CTCs, here at 7 and 13. And this 5 CTCs limit is important because this is what cell search defines as a patient being in remission or in relapse. So when a patient has more than 5 CTCs, it is considered that it's being in relapse and the therapy is not working, and the oncologist might decide for a change of therapy based on these results. However, when we look at our data, many of those patients that were classified as being in remission, they might indeed be in relapse because they present much higher numbers of CTCs. Here on the right, what you can see is also an initial testing. It is still a small cohort, and we're expanding this cohort to hundreds of samples being already analyzed, but the data is still being fully processed and put together with the clinical data of the patients. But in here, you can see a preliminary indication on the overall survival of the Ruby chip when it is analyzed, the HER2 protein in the tissue and on the CTCs. So for this Kaplan-Meier graph that you see here, in which we analyze the probability of survival for the different patients in these individual dots, once we analyzed whether the tissue expression of the HER2 versus the CTC expression shown for the HER2 as well, so when that is concordant, when the expression is consistent of the tissue and the CTCs, the probability of survival is 100% for these patients. However, for these 3 patients, as the expression was discordant in the tissue versus the CTCs, these patients had a much lower probability of survival, and that is related to the therapy that was not being according to the current expression of the tumor because of its dynamic nature. These patients were being treated with the anti-HER2 drug, but the active tumors that these patients had did not have the HER2 any longer, as they had in the first diagnosis when the tissue biopsy was collected. So this is a demonstration that the Ruby Nanomed is able to stratify metastatic breast cancer patients and also predict therapeutic guidance. Now, in here, what you see is just an analysis, the way we do the analysis with cytokeratin to identify cancer cells, pimentin that identifies epithelial mesenchymal transition cells. And this is important because these epithelial mesenchymal transitioning cells having associated to a worse prognosis, and then we use CD45 as a control to identify white blood cancer cells that might have remained in our system. Now, I have mentioned a few slides before that the limit of 5 CDCs established by Cell Search, that is this FDA-approved standard technology, was 5 CDCs. However, after analyzing our data with increased sensitivity, we identified that this threshold of 7 CDCs might indicate percentage of survival and can stratify these patients further. Some further testing of our technology, in this case, that was based on the identification of a specific antigen that is a Cia-LTN. This is known as a robust metastatic marker that we put ahead as an hypothesis together with collaborators at clinical hospitals here in Portugal. So Cia-LTN, it is an associated carbohydrate antigen, and it is expressed in more than 80% cancers. And it is also associated with our best outcomes of the disease, and also associated with a decreased patient overall survival. So if we go to a testing of the presence of the Cia-LTN in CDCs, in CDC counts, we see that in healthy donor, there wasn't any expression of STN. However, in metastatic patients, in this case, for urologic cancer patients, we see that there is 100% of expression of the Cia-LTN in the different CDCs that we have isolated. And this is one of the first proofs of the Cia-LTN quantification and identification in circulating tumor cells. Now, going a bit further, what other technologies focused on CDC analysis traditionally do is to analyze the EpCAM protein, but as we put ahead, FCN is present in more than 80% of types of cancers. So for, in this case, for metastatic and bladder colorectal cancer patients, we see in this graph that for bladder cancer patients, we have a total CDC, this is a total CDC count in black, and then in gray, we have the STN expression on CDCs, and we can see that most of the CDCs express this STN. So this hypothesis, it is the STN to be used as a universal marker in CDCs for metastatic condition. And now for colorectal cancer patients, we can also see that is in between almost 100% expression when there is a CDC that expresses this NTM marker. Now, if we go even beyond and we compare this expression of STN versus the EpCAM expression, again, now in black, the STN on the CDCs and the EpCAM on the CDCs, and you see that again, for 100% of the CDCs, the STN marker is expressed, whereas the EpCAM is expressed in below 50% of those CDCs. If we also go even a bit further and we do a genetic analysis for these metastatic bladder and colorectal patients for a specific mutation here on the right for the APC mutation in the colorectal cancer patients, and here we demonstrate how important it is to assess the current expression or the current condition of the CDCs and not of the tumors. For example, if we look at patient number 6, we see that in the tumor, there was a positive expression of this specific mutation, whereas when we go to the CDCs, there is a negative mutation, which mirrors what is happening at the metastatic site. Then if we go to patient number 3, it is, excuse me, so for these patients in gray, we didn't have the information on the metastatic side, but for example, if we go to patient 10, we see the opposite trend, that the APC was not present in the tumor, whereas in a CDC, it was positive as well as on the metastatic side. And this is again a demonstration on how important it is to have a real and a spatial determination of the expression of different proteins and also of mutations at the CDC level and not relying on the tumor condition at the first time of the diagnosis with the tissue biopsy information. And here I am presenting yet another example for gastrointestinal cancers in which we have first optimized our technology for different cell cultures of cell models for gastrointestinal cancer. So these are different cells from culture with their corresponding size and their corresponding ratio for the nucleus to the cytoplasm, which is important, as I said, for this deformability property to be able to do this isolation. So if we go and do the isolation for these different cell lines, we see that depending on the number of cells, we get different efficiency of these captures, and we also optimize the flow rate at which we have to run the blood sample to improve the capture efficiency for these cells. Still here with cells from cell lines, so models. Now, if we again study that for the different cell lines that I have mentioned a couple of slides ago, before, we have different capture efficiency also related to the ratio of this nucleus to cytoplasm. And so being the best results for the model cell line SW480. This is just to do an optimization and to have an idea of what we can expect for real gastrointestinal cancer samples. So if we go then to analyze a cohort of 11 cancer patients, as it is shown in this table, what we have assessed in this case is in green the epithelial CDCs, those ones that express epithelial markers, epithelial mesenchymal transitioning, circulating tumor cells, the ones that I said they were important for the colonization ability, for the production of micrometastases, and then in orange, the mesenchymal circulating tumor cells, those that have been reported in the literature to have a much higher colonization and malignancy potential. So in this cohort of patients, we were able to see, as you can see in the pie chart, to analyze and profile the different CTCs based on their protein expression. Now, if we want to relate this with immuno-oncology and how CTCs can be used to immuno-oncology prognosis and therapy guidance and other applications, we can find in the literature, and we can go to this very nice review then pointing to other specific works. We can see that these CTCs can be used as biomarkers in cancer immunotherapy with qualitative or quantitative assessment of these CTCs, and this has been demonstrated for several different types of cancers. We can also use these CTCs to study the evasion from immune surveillance, and so there's a lot of works on the expression of the PD-L1, which is again a druggable target, also preventing T-cell mediated destruction. We can use these CDC surface markers involved in immune escape in metastatic dissemination for examples of CD44 protein, CD47, PD-L1 that I just mentioned, and EpCAM. And we can also use this immune surveillance as well as using these CDCs as therapeutic targets. Just bring a couple of very simple examples ahead now. And so again, in this review, you can find several different examples, but you see that there are different therapies that might target CDCs and through different types of cells that are contained into this very complex micro-tumor environment. And so we can use anti-EpCAM CAR T cells to attack and reach the circulating tumor cells to avoid metastases. But also, as I said, we can do it for all the different types of markers. And there has been a lot of research into anti-PD-1 and PD-L1 for head and neck carcinoma, lung cancer, liver cancer, breast, melanoma, bladder, prostate, non-small cell lung cancer, etc. So more specifically, it hasn't only been used in research and development studies for small laboratorial preclinical trials, but also has been included in larger clinical trials. The use of CDC counts to see the therapeutic evolution of the patients. So, for example, in this paper, in which they use in this trial, they use anti-PD-L1 treatment for the metastatic bladder cancer, and what they have used is CDC counts for this specific patient, in which after the therapy, the number of CDC counts decreases dramatically. And because the CDC count is associated with the prognosis, they can use CDC counts to see whether the therapy is being successful or not. So just going ahead and having just a quick snapshot into the original challenges that I have defined at the beginning of my talk. I have spoken about the challenge of finding the needle in a haystack with the sample processing methods that can be these microfluidic devices, but then we also into the sensing technique. And so what I have shown so far, it is the use of standard immunocytochemistry or flow cytometry, so basically fluorescence-based technologies and also NGS technologies to analyze the circulating tumor cells. But we can go beyond and explore some beyond the state-of-the-art technologies. So for that, I will briefly dig into what is called optofluidics for single-cell detection. And more specifically, in the opto part, I will focus on the surface-enhanced Raman scattering spectroscopy. Long story short, SERS, that is the short for this very long name, is taking advantage of nanostructures, of metallic nanostructures, to enhance the Raman signal of molecules that are close to these nanostructures. The Raman signal is something that looks like this spectrum here, and this is something that the cancer people will be less familiar with than when we compare with fluorescence. Fluorescence is broad and might overlap, so that is impeding sometimes multiplex analysis, and also it's difficult to get quantitative information through SERS. Whereas a Raman spectrum, it is a fingerprint of molecules, and each of these individual peaks, it is a vibration within the molecule, so it offers a lot of structural information and is extremely specific beyond being multiplexing and avoiding photobleaching if we compare it to fluorescence. So this is the way nanoparticles, nanostructures that are these super specific and sensitive nanosensors look like. These are just transmission electron microscopy images of what we call gold nanostars. And then these nanoparticles can be either in solution, or they can also be put in the shape of sensors into glass, silicon, hydrogels, plastic, etc. So they're very versatile and flexible platforms. And then there is a fluidics part, and I explained a bit about microfluidics, but within microfluidics, there is a technology that is called microdroplets. Microdroplets is basically so you can see a video of these microchannels that create microdroplets, micro-contained environments that are able to isolate a species of reactions, in our case, cells, and the high throughput is huge. So we can perform 10,000 experiments per second. For what it concerns today, these 10,000 experiments can be single cancer cells contained into these nice microdroplets, microbubbles, microenvironments, and we can analyze cells at the single cell level, which is relevant for what I was explaining at the beginning of my talk related to single cell analysis and cancer heterogeneity. These micro droplet environments are like a traditional chemistry flask on a chip, and we can, as you see in this video, we can do mixing, we can do pipetting, as in this other video, we can mix reagents, and we can even have cells alive inside there and do monitoring studies and real-time studies a long time. So we have used and combined this technology, sensing technology, SERS, with microdroplets for the single cell phenotypic analysis of the protein expression at the single cell level. So for that, we have used these very beautiful gold nanostars that have been labeled and functionalized to have a specific antibody on the surface that might recognize proteins expressed by cancer cells. These are specific signatures that would be the equivalent to fluorescence, but in this case, we have super defined peaks for 3 different proteins: EpCAM by menting, the one for epithelial mesenchymal transition, and a negative control for CD45 white blood cells. So we've put these cancer cells in the presence of these SIRS codified particles and put them inside microdroplets. And what we did is to, here we have a microdroplet, here we have a cell, and to analyze the expression through the Raman signal for, in this case, 5 different cells. And we've seen that there is expression of EpCAM in these specific cells, whereas when we put cells that do not express the EpCAM, we do not have this specific peak at these specific wavelengths that we have identified. Now, we can also do multiplex detection, and in this case, we can see a profile of the protein expression of the EpCAM on these cells, single cells contained into these droplets, and also single cells expressing the pimenting, and then this is the negative control in which we cannot see this expression. So summarizing, these SERS tags might be able to substitute fluorescence markers to offer multiplexing and to also offer quantitative analysis and protein expression at the single cell level. Now, I would like to finish my talk by presenting a complex approach of a new project that we are developing in collaboration with many different partners. And what we are doing here is, excuse me, I just passed the slide by mistake, is to go beyond what I have just explained and combine different approaches. So we get the blood from metastatic cancer patients, and then we also pass this blood through the Ruby chip. We isolate the circulating tumor cells nicely and neat and there that are viable. After that, we get these viable circulating tumor cells. We encapsulate them in these microdroplets that I did show you at the individual level. And what we are doing is we are making these single cells proliferate into asteroids so that we have a clonal mirror of those single cells. And then we can do different omics analysis. We can do the growth kinetics of these cells at the individual level. We can do metabolomics. And then at the end point of these asteroids, we're going to get them out of the chips, and we're going to have traceability for the metabolomics, and then do genomics and transcriptomics of these individual asteroids. And we're going to integrate all this information into an artificial intelligence system with the clinical information to be able to decipher patterns behind metastases that might help to identify which one is the bad one, which cells and with which properties these cells, so both at the metabolic, genomic, and transcriptomic level, these cells have an effect. This is just a sneak peek into this. These individual images are asteroids that have been grown from single cells, and this is extremely difficult. You're going to be seeing asteroids in the literature that have been grown from hundreds of thousands of cells, but each of these asteroids that you see in this image have been grown from a single cancer cell in microfluidic platforms that are very robust, and now they're being analyzed for their omics, profiling. So just to finish, I would like to thank all my team at the Medical Devices Group here at INL. And then I would also, well, thank you. This is again the team, but also thank you to my colleagues and founders of the Ruby Nanomed spin-off of the research group. This is a spin-off that has raised more than 5 million in funding, and we have done a very intensive product development over the 5 years. And we will be releasing our prototype into the market next year, because we will be finalizing the first round of clinical trials for certification of the product. Without any further ado, I thank collaborators and funding and all of you for your attention. I hope this was a bit of a sneak peek into different technologies, but also how this cutting-edge technologies can have an impact on the clinical practice for cancer prognosis and monitoring and diagnosis. So thank you very much again, and I'm open for questions.
Wonderful. Thank you so much, Sara, for such a fascinating talk. And as with all the talks, we're likely to have lots of questions from the audience, and I can see that we have some questions already, so I'll hop straight into the Q&A now. So the first question that we have is, would all CTCs mirror the molecular signature of a primary tumour?
So as I did show in some of the examples, the CTCs mirror the molecular signature of active tumors much more than mirroring that of tissue biopsies that were taken at the original diagnosis time points. Now, I don't think we're in the condition, neither ourselves nor the other people studying these, to say that CDCs fully mirror the molecular signature of all the tumors, because in a metastatic setting, for some patients, this metastatic setting could be very complicated, and there might not be only one active tumor that is releasing CDCs, but there might be several. Now, it is indeed that by analyzing the molecular signature of these CDCs, we get a much more accurate snapshot of the molecular profiling of the active tumors. Hence, we will be much closer to the real situation, much closer to be able to have a therapeutic targeting that really is much closer to what is really happening on the patient than if we use a tissue biopsy, if that answers your question, but feel free to ask for further explanation.
Perfect. Thank you so much for that. And I think we've got time for one quick question before we're out of time here. So the final one, how can CTC-derived spheroids help towards the identification of immuno-oncology targets?
So thank you very much for the question. Again, that is extremely interesting. So single CDC-derived spheroids, as I said, would be a very particular yet interesting model for the identification of immuno-oncology targets, because when we have patient-derived spheroids, it's often coming from several hundreds or thousands of cells. And these several hundreds of thousands of cells will contain many different clones in the analysis, against oversimplifying. And these many different clones might be difficult for the identification of a specific immuno-oncology target. However, the single CDC-derived spheroids these spheroids will mimic the clonal condition of the original single CDC. And this original single CDC would contain the specific target for the immuno-oncology therapy. But because we have the spheroids, we would avoid amplification methods for genomics and transcriptomics, which often include errors during this amplification and noise into the analysis for this targeting. Also, having these single-cell patient-derived spheroids in robust and high-throughput platforms can streamline the testing of these therapeutic targets that pharmaceutical companies are developing and are including in phase 1 and phase 2 trials. And also, last but not least, for these immuno-oncology and therapeutic targets, by having these single-cell-derived spheroids, we could also, the pharmaceutical companies and the clinical trial and CROs could also better select the patients that might go into a specific trial. So if they're selecting, if they need 200 patients and they select them and include them in the trial without knowing whether they might contain that therapeutic target or not, many of those patients would not benefit first, and second, the clinical trial efficiency will be much lower because the patients that might have been the target have not been included, and some others that are not have been included. So pre-selection of patients for clinical trials is also very good use for these single-cell patient-derived spheroids before those patients are included in the trials.
Wonderful. Thank you so much, Sara, for your fascinating talk and your excellent answers. Unfortunately, that's all that we have time for today, but any questions we didn't have time to get to will be sent to Sara after the event. As with all of the other talks in this event, this talk will now be available to view whenever you want for the next 30 days. And that just leaves me to say thank you so much again, Sara, and we'll be back soon with the next talk.
Thank you very much.